All right, so what I want to do today is talk about what we've learned from basically from comparative genomics about the diversity of bacterial and archaeal genomes in particular, just the big picture, and how we compare genomes a little bit, and a little bit about differences between bacteria and archaea and eukaryotes. And really, this is sort of basically, I don't know, 6 or 7 main topic areas for the diversity and variation in genomes within bacteria, within archaea, within eukaryotes, and between them. And one that I don't know how much people here sort of will appreciate, but many people in the field who've been around for a while were pretty surprised by a lot of this. So in most textbooks, and most people in the field had this notion that bacterial and archaeal genomes were predominantly made up of a single circular chromosome, so a piece of DNA that's covalently linked into itself. And most eukaryotic chromosomes are linear. And then occasionally, a few species would have a small or multiple small pieces of circular DNA. And these were called, this big piece was called the chromosome, and these little pieces were called plasmids. And that's how E. coli is. So of course everything is going to be like E. coli, and we can, base all of our textbooks on how E. coli works. And it turns out that there's just much more diversity and variation in the general structural diversity of how genomes are arranged. To the point, and I'll talk about this in a minute, it's no longer straightforward to just look at the genome structure of an organism and say, this is a chromosome and this is a plasmid, which people did for many, many years. And if you look at this, this table I made many years ago, but if you look at, there are some organisms where there's a plasmid in this, or a second genetic element in this Streptomyces that's 356,000 base pairs long. In Agrobacterium tumefaciens, there's a circular large piece that's 2.8 million bases long, and then there's a linear, so non-circular large piece that's 2 million base pairs long, and then multiple other elements in descending order in size, shown here, Deinococcus radiodurans, 2.6 million, and then 412,000. And many of these second elements in organisms are larger than the entire genome of some other bacteria. And so it's just, what are these things? Should we call them chromosomes versus plasmids, and what does a name even really matter? The main thing to know is that there's lots of diversity in the general arrangement of the chromosomes and DNA elements in organisms. There's much more linear DNA in bacteria than was previously expected. The model was that everything is going to be circular. It turns out that some organisms have linear, everything is linear basically. And linear, we didn't go into the details of this, but one of these big differences between most bacteria and archaea versus eukaryotes is in how they copy their chromosome. It's complicated in organisms that have ends to the DNA because of the mechanism by which DNA is copied. And organisms with linear ends have to have some special adaptation to continually basically extend the ends of their chromosomes so that they don't lose pieces. And in humans, there's an enzyme called telomerase. And this enzyme is basically continually adding these special little caps onto the end of the chromosome, and those caps get lost every time you replicate the genome, but it keeps adding caps back on. There's a lot of interest in telomerase because it's thought that degradation of the chromosome ends or decrease in activity of telomerase might be related to aging. And so for sort of many years, people considered this a hallmark difference between eukaryotes and versus bacteria and archaea, and it turns out it's not true. And in the bacteria that have linear chromosomes, they also have to have some adaptation for copying the ends. And it appears that these are separate convergent inventions of the process to maintain the chromosome ends. And if you look across eukaryotes, for example, some organisms, I think Drosophila actually, instead of using telomerase, uses a transposable element. Some organisms just keep sort of copying a transposable element to the end of their linear chromosomes. And there's sort of a diversity of adaptations. And anyway, this is important because a lot of sort of the default understanding of the function of bacteria and even drugs that target bacteria relate to the mechanisms of replicating the chromosomes. And it's very different when you have a circular chromosome versus a linear chromosome. So if you look, now there's probably even more diversity seen in bacteria and archaea than is shown here, but I think this sort of captures it. Whether or not you call something a plasmid or a chromosome, I don't think makes any real difference. But what's important to think about is that there are some genetic elements in organisms that carry lots of essential genes or sort of core housekeeping genes that are essential in all conditions. That's what we tend now to call chromosomes. And then genetic elements that carry genes that are only required in sort of specific conditions like resistance to antibiotics or resistance to toxins or production of some chemical, those now tend to be called plasmids. So if you don't have many universal housekeeping core genes on a genetic element, they tend to not be called chromosomes anymore. And it's just sort of a simple way of distinguishing when you talk about the genetics of an organism to refer to the element that has the housekeeping genes and the element that doesn't. And so if you look at this table, for example, Vibrio cholera, when the genome was sequenced and I was part of this project, they discovered that there were two big circular pieces, one about 3 million base pairs, one about 1 million base pairs. 95% of what you would call housekeeping genes are on this 3 million base pair genetic element. Like 2 of them are on the other element, but a decision, there literally was a meeting among the Vibrio cholera and Vibrio experts to decide whether or not to call this second thing a chromosome or a plasmid. I was not involved in that discussion, but because it had some of these sort of housekeeping genes, it's become known as a chromosome. Anyway, plasmids themselves, again, whatever you want to call them, smaller genetic elements in organisms, or these elements that are not carrying a lot of housekeeping genes, tend to have lots of really interesting functions encoded on them. You can view them as sort of conditional adaptation encoding genetic elements. And so there's lots of plasmids that encode antibiotic resistance or resistance to other type of elements like toxins. There's some that are involved in mating, so carrying out sort of movement of DNA between organisms. There's quite a few of them that encode virulence and pathogenicity factors. There's another complication, which I'm not going to talk probably at all about, which is that many of the plasmids that you see in different bacteria in particular and archaea have very close evolutionary ties to viruses. And so there are viruses that many of the genes encoded on bacteriophage are also found on many plasmid elements. And it's unclear what the whole history of this is. It may be that they're exchanging DNA with each other. It may be that plasmids represent captured viral genomes. In some sense, there's a virus, it has a genome, it infects a cell, it loses the ability to make the protein coat, but the DNA is still there and it might get transmitted, yeah.
It'd be like when they insert their genome, it'd just be like, it doesn't really get attached to the bacteria genome, but gets ligated at the end in a circle.
Yeah, so you have a virus that has its own DNA in it. It comes and some bacteria has its own genome. It injects its DNA into the cell, and there are processes by which that DNA is then integrated into the chromosome. And the same is true for plasmids themselves. They can get taken into the cell, and they're frequently integrated into the chromosome. But they don't always get integrated. And so when a plasmid does this, one way to distinguish when you have sort of pieces of DNA, if you're sequencing a genome, between, say, something you've never finished the genome of, like you can't figure out where it goes in the chromosome, versus a free plasmid, is that plasmids have these suites of genes in them that code for their ability to get replicated and segregated into offspring cells to get maintained. And in viruses, you don't always see those because their sort of mechanism of replication is either to make copies themselves into the viral particles or to get integrated into the chromosome. So yeah, I mean, I don't know examples off the top of my head where viruses that infect cells, sometimes the chromosome goes in, sometimes their DNA goes into the chromosome and sometimes it doesn't. I don't know if Holly knows any examples of this, but they probably exist.
I thought Agrobacterium had a correspondence like that.
So Agrobacterium has, some strains of Agrobacterium have large accessory genetic elements. One of them is called a TI plasmid. It induces tumors in a variety of plants, but it's not there's no viral particle. It's just a piece of DNA that can get injected into plants and lead to tumor formation.
What's the transmission vehicle?
Just like a chromosome. It gets copied by Agrobacterium during normal growth, and then when it infects plants, it forms some type of tunnel, basically, into plants, injects that piece of DNA into them. That DNA then takes over the machinery of the plant to create these tumors. And it actually can get integrated into the plant genome itself. And that's why Agrobacterium and its relatives are used for genetic engineering of many plants, because you can modify Agrobacterium in the lab, change the DNA that's in this tumor-inducing plasmid, keep the part that is required for it to inject into the host, and get it to transform a plant cell. But there's no viral particle here. It's all plasmid replication. And the distinction, I mean, it's a really gray area. There are, transposable elements that also get sometimes encapsulated into viral particles or protein coats. And, there are many cases where it's sort of hard to say if something is solely living its life as a plasmid or participating in some type of viral replication and so on. But many people are really interested in these, if you're interested in the diversity of a group of organisms, let's just say Agrobacterium, and you wanted to quickly discover parts of Agrobacterium that are variable among strains. A good way to go after it would be to go after the plasmids, because they tend to be the sites for which organisms have sort of lots of diversity and variability. It's not that you don't see variation in the chromosomes, but it's sort of a simple way to target novel, interesting functions. So genome size is another one of these issues that, as people have done more and more genome sequencing, the ranges of genome size expectations for different organisms have changed quite a bit. This figure from maybe 8 or 9 years ago showed the range for eukaryotes, which is enormous. So this is, 1, 2, 3, 4, 5, 6, 7 orders of magnitude variation in genome size, and there probably even stretches a little bit further. But here are, amoeba, which has a genome size 10 times as big as humans, and there are some other eukaryotes, I forget what this is, that's almost 100 times the genome size of humans. Bacteria have now been found to have, a few of them have genome sizes of something like 15 million base pairs, so bigger than a few yeast and fungal genomes. There are bacteria that live as intracellular symbionts inside other organisms. They never sort of live as free-living organisms, so they're kind of like mitochondria and chloroplasts almost, but there are some of them that have genomes that are on the order of 100,000 base pairs, so really, really small. So there's a bit more variation in genome size than maybe is shown here, but it's still, there are no bacteria with massively large genomes like you see in eukaryotes, and there are no Archaea with massively large genomes like you see in eukaryotes. So I mentioned this before, but the difference, so the reason that some eukaryotes have these massive genomes is mostly not that they have many more genes than are seen in either the other eukaryotes with smaller genomes or even in bacteria and archaea. It's mostly that they have lots of intergenic DNA in between genes, or in between protein coding regions of genes. So I'll come back to that figure in a second. If you just do a plot of the genome size versus the number of genes, you see a very nice relationship for bacteria and archaea. As you get larger and larger genomes, you basically get something on the order of 1,000 protein coding genes per million base pairs, or 1 protein coding gene per 1,000 base pairs. But then when you get into eukaryotes, it's just really scattered, and there's basically there's a little bit of a relationship, but not a very good relationship between genome size and the number of protein coding genes. And the reason for this is, pretty simple. There is, here in humans, it's showing, here's a protein coding region, or I think it's just a exon, in 50,000 base pairs. And in this region, there's basically one gene with a bunch of different exons. And in E. coli, in the same region, there are 50 protein coding genes in the same total amount of DNA. And so you basically have the genome filled up with other stuff. And there's, some people may be aware of this, there's a big ongoing debate about what that other stuff does in eukaryotic genomes. It was originally labeled junk DNA by a lot of people before anybody ever actually looked at it. And it is true that when people first started to look at the rest of eukaryotic genomes, they found lots of repetitive DNA. So you find a lot of transposable elements, for example, filling up the rest of the genome of eukaryotes. There are these little miniature elements called line elements, for example, in the human genome. There's something like 200,000 of them, or more than that, in the human genome, and it makes up like 20% of our DNA. Something crazy like that. And so, sort of understandable that when people first started to get genomic data or look in more detail at genomes of eukaryotes, that they said, okay, here's the interesting part. It's the protein coding regions. The rest is just junk. We're going to ignore it. It's unclear whether or not that was a smart thing, I think, still today. There clearly are regulatory functions in much of this sort of spacer regions in eukaryotic genomes, regulating transcription, regulating chromatin structure, regulating methylation status, regulating a variety of aspects of the genome. There are non-coding RNAs that are now being discovered in many of these regions of the genome. So, yeah.
So, bacteria have transposable elements as well, but they tend to kick them out, right? So do their transposable elements have special adaptations that keep themselves in?
I think for most purposes, you can view transposable elements that are present in a genome as being somewhat selfish. I mean, they're not independent of the cell or the organism in which they live. So they're not completely selfish, Darwinian, evolving entities. But there are many aspects of their life where you can think of them as having selfish tendencies. And their tendency is to make copies of themselves, right? They're transposable elements, and some of them, well, some transposable elements actually, they duplicate themselves into another region of the genome. Some of them cut themselves out and move to another region of the genome. The duplicative ones, if there's no control on them, will spread throughout a genome. And as far as I'm aware, the model to explain why you see a lot of these in eukaryotic genomes and many fewer of them in bacterial and archaeal genomes is that there is, in essence, a selective pressure against large genomes. And that there's, once you cross over a certain threshold of genome size, that's detrimental. Much more detrimental in bacteria and archaea than it is in big eukaryotic cells. And I don't know, I mean, it may be size constraints, but there's clearly some constraint upon massive genome size in bacteria and archaea.
There are transposable elements in bacterial.
Absolutely. So there are transposable elements.
How do they stick around?
Right. So again, if you think of them as somewhat selfish elements, you can imagine, sort of 5 different populations of bacteria that start out identical to each other, each with one copy of a transposable element. If in one lineage the transposable elements go crazy and their transposase activity gets really high, they're going to scatter throughout the genome. They will insert, first of all, in bacteria and archaea, if they insert somewhere, they're likely to insert into a gene because that most of the genome is coding. So that's already going to have potentially detrimental effects, although it could also have beneficial effects. But if they go really at a very high rate, that lineage is going to do poorly because the genome is going to get bloated and there'll be selective pressure against that genome size. And so what probably happens over some period of evolutionary time is that there's selection against rampant transposable element activity for the transposons that exist in bacteria and archaea. And so they're there. They can make copies of themselves, but they're not as active as the transposable elements that you see in some plants and animals. I mean, they do occasionally go wacky. And they do another thing, which is really important. So if you have a sequence here, and you go through some copying of itself throughout the genome, these copies now serve as sites for recombination, and you can get intragenomic recombination between them. And that can lead to either duplications, depending on the orientation of these copies, or deletions, where a region of the genome gets deleted out. So repetitive DNA, which can be created by transposable elements, can lead to chromosomal instability. And so that's another sort of important factor to consider when a transposable element moves around a lot. There's probably, lots of people study this. I don't know all the details. There's probably lots of subtle selective pressure against. So if it inserts in a certain way where it's flanking, closely an incredibly important gene, and that would be prone to deletion, those lineages are going to get weeded out, right? So, but in many plants and animals, the transposable elements are not exceptionally active at any one individual time either. There's sort of periods of bursts of transposable element activity. So a classic example is sort of production corn versus more wild landrace corns, where the theory is that many Native Americans selected certain types of corn because they had lots of variation in color patterns. Those were caused by transposable elements. So if you select for variability, in eukaryotic genomes, one of the first things that you end up enhancing is the activity of transposable elements, which are suppressed most of the time, or they would be dead too.
Yeah.
So I think mostly it's just there's a cap on genome size, and anything that leads to lots of extra DNA will eventually be selected against. And one thing that will do that is lots of copies of transposable elements. Does that sort of answer? So I mentioned this a long time ago in the class, but one of the sort of fundamental structural differences between bacteria and the archaea on the one hand and eukaryotes on the other is not just that the genes are much denser in bacterial and archaeal genomes, but you actually see this sort of really fundamentally different process where you get these entities called operons. And what happens with an operon is you have a piece of the genome, the DNA. RNA polymerase makes a transcript, copy of the DNA, starting at a promoter site, and you make a transcript. And inside this transcript are the protein coding regions of multiple proteins. And so you can have somewhere inside this transcript there will be a start site and an open reading frame here, and you will translate that into protein 1. And then there'll be another start site, and you will translate that into protein 2, and another start site, and you'll translate that into protein 3. And this whole region is called an operon. And the reason, well, I'm not sure whether it's the reason. What happens is basically that this is going on inside a bacterial or archaeal cell. The RNA gets made, and just even as the RNA is getting made, the ribosomes bind to the RNA and start making protein. And this is what I mentioned before, that coupling of transcription and translation that occurs in bacteria and archaea. And in eukaryotes, the RNA is made, and then for protein coding genes, it's transported out of the nucleus. And the translation is outside the nucleus. And in most cases, although there are a couple of exceptions, there's one RNA, in essence, for each protein coding gene. You don't see these stretches of one RNA getting translated in multiple pieces into multiple protein coding genes. There are a couple trypanosomes, have these long, they came up with a new name for them. I think they're called Polycistronic Rnase, but they basically do the same thing, yeah.
So is there like some other enzyme that kind of like cuts them up so that they don't get all like made into one big weird protein thing?
Here, in bacteria and archaea?
Yeah.
No.
Oh.
So
I think I remember reading it, that they kind of like are part of the same kind of thing.
yeah, so this RNA will get translated at once into, there'll be a ribosome binding here, a ribosome binding here, and a ribosome binding here.
Oh.
That's the machinery for doing translation all at the same time on one piece of RNA. And you actually see, I don't have the electron micrograph, but you see these amazingly cool pictures because there'll be, protein will start being made. It'll be short when it's right near the beginning. And there'll be another ribosome here, and another one here, and another one here, each one with a slightly longer growing protein coding chain as it's going through the RNA. So you get these, I think they're called Christmas tree structures, because you see these longer and longer sort of globs of protein coming off the RNA, still attached to the DNA. I mean, it's still basically, the DNA gets made into the RNA, and as it's, as you're going along, let's say there was another protein that was supposed to be made here, the RNA polymerase could still be making this RNA. as it's making it, the ribosomes are making protein off of it. And this allows for a really cool regulatory process that only exists in bacteria and archaea, whereby what you're making, the protein that you're making, and the codons that you have for the protein that you're making influence the rate of transcription of making RNA off of the DNA. So there's a direct linkage between what protein you are making and how it's working and the rate of actually making the RNA. The classic example of this is in the genes for making, for the trip operon in E. coli, where if you don't have a lot of tryptophan in the cell, the tRNAs that are supposed to be charged with tryptophan, if you have enough tryptophan, you should have tRNAs that have the opposite of the codon for tryptophan. They should have the amino acid at the end of them. That's how proteins are going to get made. If you're starving for tryptophan, those those tRNAs don't get made. Basically, I mean, you have the tRNA, but it has no amino acid at the end of it. The cell will pause as it's trying. If you make certain RNAs that are supposed to code for a protein that's supposed to put in tryptophan, the process will pause at those sites. And there's a regulatory process that says make more RNA for the trip operon that's going to make tryptophan if you have that going on. And there's hundreds of these in bacteria and archaea where you can sense something going wrong in making a protein and directly regulate transcription. And eukaryotes can't do this because proteins are made outside of the nucleus. So it's a sort of regulatory process that's completely different between bacteria and archaea on the one hand and eukaryotes on the other. Eukaryotes tend to have regulatory processes where there's sensing out in the cytoplasm or associated with the ribosomes, but then they have to do a signal transduction process to send information back into the nucleus to regulate transcription. So for example, steroid hormones, one of the things they do is they get into the nucleus to regulate transcription. And there's lots of processes in eukaryotes where sensing signals are made in the cytoplasm to be sent into the nucleus to regulate transcription. And again, bacteria and archaea don't have to do that because there is no second membrane blocking off the DNA. Does that sort of make sense with what's going on? And this is just one example. There probably are 5 to 10 major sort of classes of regulatory processes that either are only in eukaryotes or only in bacteria and archaea because of this coupling of transcription and translation. And there are some, I don't have pictures of it, but actually I have one in a minute. There are some operons in in certain bacteria that are, 30 genes long. You can make this gigantic RNA and have more than 30, hundreds of ribosomes attached to it, making protein off of it. And another really important thing about bacteria and archaea versus eukaryotes, this is one of those other regulatory processes, is that because you're making protein off of the RNA that is itself basically close to the DNA, there can be all sorts of weird effects when you make proteins that interact with DNA. So if you make a protein that is a restriction enzyme, for example, that is supposed to chop up DNA, because bacteria and archaea, because they do this coupling, those enzymes are going to be right next to the DNA. Whereas in eukaryotes, you're going to do all your synthesis out in the cytoplasm. The DNA is much safer. And so there are a bunch of other regulatory processes that are there to sort of prevent DNA binding or manipulating enzymes from either being active when they're first being made off the ribosomes or from interacting with DNA in the neighborhood of their own gene. And these, again, something that just doesn't happen in eukaryotes and is really different with bacteria and archaea. Okay, now switching, so that's sort of just the big picture was gene genome sort of content, genome size, gene density, gene sort of neighborhood arrangement, and now going to talk a little bit about just overall gene content between genomes. And this was pretty surprising when people first started to show this. So Tiger, we mentioned this before, Tiger sequenced the first genome of any organism in 1995. This Haemophilus influenza. Shortly thereafter, they sequenced the genome of a second organism. At the time, they thought it was the organism with the smallest genome available, this Mycoplasma with 470 or so genes. And they compared the Mycoplasma genome to the Haemophilus genome. And in fact, a bunch of other bioinformatics people did this over the next couple of years. And they all basically got the same results, which is that they share something on the order of 250 or so protein coding genes that appear to be closely related, not closely related, but orthologous between Haemophilus influenza and Mycoplasma, sharing a common ancestry. And there were all these papers written and lots of work talking about the minimal genome of an organism based upon this comparison. That this was, we compared two genomes. The minimal genome for bacteria is going to be about 240 genes. This is the core essence of bacteria. And then people sequenced the next genome, and the core got a little bit smaller. And then they sequenced the next genome, and so on. And when this figure, I think, came from about 25, first 25 bacterial genomes, they only share 80 genes in common. And as people have done more and more bacterial genomes, I think the list is down to about 20 now. 20 protein coding genes or large non-coding ribosome RNAs that are shared across all these genomes that appear to be homologous. And this is, annoying, vexing, let's put it that way. And the explanation for this is not entirely clear when it was first observed. I mean, there were basically two models. One was that sequences might be evolving for certain genes at such a high rate that we can't detect homology between all of these diverse organisms. And, well, three explanations. So sequences are evolving at a very high rate. Two, which is a pretty reasonable idea, but turns out to be wrong, which is that the annotation was so bad that we just couldn't compare across these genomes. That's definitely not the full explanation. And the third, which appears to be correct, is that even though at any one time, hundreds of genes in an organism appear to be required, over billions of years of evolution, even genes that we think are absolutely essential now, like ribosomal proteins, they can get lost. They don't keep everything. And organisms will replace those genes with something else. They may need to replace them with something that carries out the same function, but it may be convergent evolution. You may not have any sequence similarity between two genes that carry out some core function that occasionally get replaced. This is now known as non-orthologous gene displacement. Don't worry about the details of that name, but basically the model is that you have organisms evolving over time, and there's basically a slow but reasonably steady rate of invention or addition of genes and deletion of genes, even key, genes that we think are, might be really important. And the probability that a gene is deleted is highly variable, might be low for housekeeping genes and high for sort of accessory, non-important functions. But it's high enough for even sort of core housekeeping genes that if you average over 25 genomes, you're going to see very little shared across all those genomes. And this, I think, should have prepared people for the next figure, but probably did not. So people were getting a growing appreciation for the variability among bacterial genomes that were mostly distantly related organisms. I mean, Haemophilus influenza is a gamma proteobacteria. In one class within one phylum. Mycoplasma is in the Firmicutes phylum. It's related to Streptococcus and Bacillus. They're actually really weird organisms. They've lost their cell wall. So they're in the gram-positive lineage, but they don't stain gram-positive with this gram stain because they've totally dumped their cell wall, but they're, billions of years separated from Haemophilus influenza. And many of these other genomes were also, really novel and distantly related. So people, had this model that this slow ticking of gene loss and occasional invention of genes would explain this pattern. And then a couple of E. coli genomes were sequenced after the first initial E. coli genome. And the people who worked on those genomes made this figure that is shown in virtually every study now of bacterial evolution, which is that if you compare 3 E. coli genomes to each other, so these are the same species, those 3 genomes are incredibly similar for a subset of their genes. In this case, about 3,000 protein-coding genes shared between these 3 genomes. Each genome has, however, about 5,000 protein-coding genes. So even though they all share 2,000 genes with each other, they each have many genes that are not shared across all 3 of them. Some shared with number 2, some shared with number 3, and some unique for each organism. So what could explain this? I think, we have a very good model for this, but I'm going to walk you through how people figured this out, which is the first thing that they did, or the best way to understand this is to first look at these shared genes, these 3,000 genes that are shared between the 3 E. coli genomes. They're virtually identical to each other. At the DNA sequence level, they're 98, 99% plus identical to each other. So they're basically the same genes with a couple of polymorphisms. What people then asked was, what is the order of these genes between the different genomes? And the way you do this, the simplest way to do this is with what's called a dot plot. I don't know if people are familiar with this, but I took out the figures. If you want me to go through this, I can. You basically take a stretch of DNA, or in this case, let's just do it with the phrase from Shakespeare, and you plot it against itself or against another phrasing with the x versus y axis. So here's to be or not to be versus to be or not to be. This is what happens when you have a repeat in a region. So you do a dot plot. There's everything dots on the diagonal because 2 lines up with 2 and b lines up with b and so on. But because there's a repeat, you see another dot here for 2 versus the ending 2 and b versus the ending b. And then here's another example, A, B, C, D, E versus A, B, C, D, E. So a duplication versus itself. So you see the diagonal, and then here's one duplicate versus another duplicate. And then here is another case where I've added a little insertion in one region versus here. And then you can play around with what this looks like. So here, A, B, C, D, E versus A, B, C, D, E, and then it goes up versus A, B, C, but there's a little jump in the second repeat to D and E, and then here's the Y and Z lining up with each other. So you can sort of just imagine what it would look like if you did a dot plot now of all the genes in a genome, just like we're doing with these words or these letters here. Another thing to realize is that when you do this, and I'll show you an example of when you do this poorly. So something we have not mentioned. Most bacterial genomes that are circular, the way they get copied is there's something called an origin of replication. And the copying process starts at this origin. Remember, this is double-stranded DNA. And the cell will actually copy in both directions from this origin of replication and start copying the genome. You basically get little bubble where this part is duplicated as the DNA polymerase sort of travels outside around the circle. And it gets bigger as you continue to replicate. And eventually you get to the end, which is called the terminus of replication. And we'll come back to this in a minute, but this origin replication is actually an important way to orient genomes relative to other genomes or to itself. So when you do these dot plots, people frequently try to have the origin of replication at over here, and then you go around the circle. The terminus is going to be halfway down here. And then you continue around the circle back. Basically, this is the other side of the origin of replication. So if you do this with two very closely related genomes, it'll look like this, right? So there's going to be just spots along the diagonal. All the genes line up in the same location as their homologue in the other genome. And if you do this with E. coli K12 versus O157H7, so that was this genome and this genome. They share, these 3,000 genes across all of them, but if you add them up, they share about 3,500 genes with each other. But O157H7 has, something like 1,500 genes not in the other one. And K12 has something like 600 genes not in the other one. So if you line them up with each other, all the genes versus all the other genes, you see mostly everything is along a diagonal. You see this little gap here, and then mostly things on the diagonal. Then here's an inversion where the genes are in an inverted order relative to the other one. And you go along, they're basically all roughly in the same orientation between these two genomes. And what doesn't show up really well here, because the genome is really big, and there are lots of genes, is there are many cases where there's a little gap. Here's another one. A little translocation, little jumps in this alignment. Those are cases where one of the genomes has many genes just completely absent in the other genome. They're called islands now, little islands of DNA present in one genome and not the other. And what's basically going on here is now mostly the model to explain this is that you have an organism. It separates into two lineages. Those, in this case, could be two different strains within a species. They could eventually be different species. And what happens is mostly you get occasional point mutations in individual genes that start to differentiate the two genomes from each other. But occasionally you get insertions of large stretches of DNA into one strain and not the other. Or so if we compared this to this, there would now be extra DNA in, say, K12 versus O157H7. And if we did a dot plot lining them up, you'd see that they lined up really, really well, except there'd be this little jump in the alignment where this had some extra DNA that the other one didn't have. And occasionally you get a deletion. So maybe this section of the genome gets cut out, ligated together eventually. And then you do the dot plot. You'll still see everything lining up really well, but now there'll be the inverse. It looks like an extra piece of DNA in O157H7 compared to that one. So these two events, deletions of regions of the genome and additions of regions, little bits of DNA, create this. The massive differences in the presence and absence of genes, even among 2 strains, 3 strains of the exact same species. So this is a quarter of the genes in O157H7 are not in K12. Yeah?
I don't know how important it was to understanding the concept. I was a little unclear about an inversion around the terminus version.
We'll get to that. Yeah, that's the lot, yeah. We'll come back to that in a second. So does this basically, this part so far make sense? So this is not, I mean, people had an inkling of this, but the extent of this was really, really surprising. And it basically means, it means that the rate of insertion of DNA into new genomes or deletion of DNA from bacterial genomes is much higher than anybody expected, and much higher than what you see in plants and animals. So if we compare all of us to each other, we're going to basically share 99.9% of all the protein code, probably almost 100% protein coding genes and all the other non-coding RNAs with each other. And this is 2 strains of E. coli. They are the same species. They share these 3,000 genes that are virtually identical to each other. They have all sorts of functions that are E. coli functions. They can be really considered the same species, but they have massive polymorphisms. Yeah.
I don't know if I really understand this correctly, but you know when we were talking about before about sequence alignments, how do you figure that out if you can't even like tell if it's been deleted or added or?
For a whole genome, you mean?
Yeah.
Can we wait 1 second until I show this X alignment thing and come back to that?
Yeah.
Because I can show you how that was actually done. Yeah.
So has anyone done a survey where they went through different sort of species of like bacteria, Archaea, and like sort of found the range of diversity from like, I don't know, maybe E. coli has a particularly diverse species.
Yeah, so the first example of this was actually done in Streptococcus.
Mhm.
And some people from the place that I used to work, I wasn't involved in the study, that did a survey of about 10 strains of Streptococcus. And they made a little plot, which was the number of genomes that you looked at versus the total number of genes. And they did a rarefaction curve, just like with the number of samples versus the number of species, the exact same thing. And if the diversity was low, you would expect asymptoting early. And what they saw was very little detectable asymptote. And they came up with a term called the pan-genome to describe the genetic diversity within a species, the massive number of genes that you would see within a species. And I am recording this, but I'm just going to say it, they made the ridiculous conclusion that this meant that there were infinite genes within Streptococcus. Now, they had only sampled like 10 genomes. They had no idea if this was going to asymptote, when they hit 40, or they didn't even do, like a chow, right? I mean, like, you couldn't even estimate the total number from this small of a sampling in some cases. So as we're getting more and more genomes, people are starting to get a better idea for the total gene set. And what you see is within a lot of species, like E. coli, we have yet to reach any close approximation of an asymptote for the total genetic diversity within a species. But most of the genes that are being counted in these surveys are found only in one genome. They're predicted genes, in fact. They're not even known to be real genes. So you sequence many genomes, and occasionally in one of them, you find 10 genes not in any of the others. Well, that's predicted genes. How many of those are real? It turns out many of them are spurious. So if you plot this with genes that are found in 2 genomes, it's much lower. So the genetic diversity, there's no doubt, in some species is massive, and in other species it's lower. But I have not seen a detailed comparison, because it's only been in the last 2 years that people have started to sequence the pan-genome from many species. So we have E. coli, we have a couple of pathogens, we have basically no pan-genomes from non-human pathogens even now. So I don't think we know. whether or not there's going to be, yeah.
Are you saying that this lack of an asymptote is in part driven by the craziness of just one genome within a sample?
Yeah.
Okay.
And what you really want to do, although I haven't seen anybody do this, is it may exist, I just haven't seen it, is build an evolutionary tree of the group and sample, from across the evolutionary diversity and ask what is sort of, if I sample across the evolutionary diversity, do I see something different than if I sample, say, 10 things from just this branch? And that will tell us more about sort of the mechanism by which this diversity is generated than we really know right now. So the diversity is enormous within bacterial species. Let's just be clear. It's a little bit lower when people have done these surveys of Archaea, but it's still pretty high.
But couldn't that just be an aspect of the fact that we haven't really sequenced Archaea?
Yeah. This is the cutting edge now of questions about microbial diversity, is to try and understand where all this new DNA is coming from and what is maintaining it in populations and whether it has any functional consequences. Clearly some of it, a lot of the pathogenicity and virulence factors are in the regions of the genome that are unique to O157H7 compared to K12. I was just going to show, this is, if you compare all those genomes in that plot with the 82 shared genes, most of the shared genes are things like ribosomal proteins. And over 3 billion years of evolution, they've even maintained the same order between many of these organisms. So there is a massive constraint upon the order of genes in these operons for ribosomal proteins. Okay, now coming back to the genome inversions, and I can't remember, what was your question about?
The sequence alignment.
Yeah, the sequence alignment. Okay, so when I was working at this institute, this exact issue came up when we started to generate the first genomes where there was a close relative that the genome was available. So if you didn't have a close relative, so if you do a dot plot of Helicobacter pylori versus Haemophilus influenza, they're in proteobacteria, but they're in different classes. You don't see anything. You do this plot of these are the protein coding genes. There's no obvious pattern of any conservation, although this, I think, is the ribosomal protein cluster, where there's a tiny bit of conserved gene order between the organisms. So there wasn't really any way to test how well you could actually line up genomes because they were so distantly related that there was no detectable alignment. And when the Vibrio cholera genome was sequenced, this was sort of one of the first opportunities for comparing genomes of organisms that are different species but are reasonably closely related to each other. And the question, came up was, how do you actually align two genomes to each other as opposed to two individual genes? And there are really two main strategies for doing this. And both got used. So when people were comparing the E. coli genomes to each other, they were so closely related to each other that you could actually do what's called a DNA alignment. So you could take the entire genome of one E. coli and start to slide it against the entire genome of the other E. coli and find out where they lined up well. And then there would be regions where they didn't line up well, and you would have to run some alternative analysis of those regions. But when you lined them up, the regions that lined up well, you could actually do this literally by hand. There were regions that were 100% identical, and then occasionally you'd see a little polymorphism. And the big differences were either these big insertions in one of them, or deletions in the other one, or occasionally an inversion. And so people basically adapted the programs that were used for lining up two gene sequences to extend those to entire genomes. One of the people who did this was Aaron Darling, who did it before he came to my lab, but he used to be He then worked in my lab for a while. He came from the big E. coli genome sequencing lab. He developed one approach to doing this. And this guy at the institute where I worked at, this guy Steve Salzberg, developed another approach to these DNA, whole genome DNA alignments. And they basically were taking sort of the same concept as you would do with individual genes. But there's an alternative model which turns out to work much better when you get slightly more distantly related organisms, which is to break the genome up into chunks, like individual genes, and then search those individual genes against the other genome and find matches. You can set a threshold and say, give me everything that has 80% identity between my gene and genes in the other genome. And then you can plot the location of those genes in genome 1 versus genome 2. And that's what this figure is. So this is taking Vibrio cholera, the big genetic element in Vibrio cholera, and the main chromosome in E. coli, taking each protein coding region in Vibrio cholera and searching it against E. coli. And then if there's a match, plotting the location of the gene in Vibrio cholera versus the location of the match in E. coli. And this is basically a way you're not aligning the chromosomes to each other, but you're looking at how conserved the location is of particular genes in the two genomes. And again, that's how this was made. So it's a really quick way to see that these two genomes are very, very similar in the order of genes that are shared between the two genomes. And you can take that information and guide a DNA alignment then. Back align the DNA level. Now people use these whole genome alignment algorithms that I'm happy to try to explain how they work, but basically it's sort of the equivalent of these individual DNA alignments. But back at this time, those were not very robust, and we did, pretty much everybody did these all versus all protein coding region versus protein coding region alignments. And so you see this thing, if you do this plot of Vibrio cholera versus E. coli, it basically looks like a mess, right? Whether or not you can... Ooh. All right, so it looks like a mess, and one reason for this is that there are many genes in each of these genomes for which there are many homologs in the other genome. So there are protein families like transporter proteins, where Vibrio cholera has 100 members of some protein family, and E. coli has 200. And for each one of those genes in Vibrio cholera, you're going to plot hundreds of little spots for it in E. coli. And that just makes this a mess. And so if you take this and then filter it out and say, just show me the best match, the highest, most similar match between E. coli and Vibrio cholera, you start to see some, we call this the angel of death pattern. I don't know if you can see it from over there. And if you then plot this only if the genes are like basically close relatives, orthologs, it's not there. We realized that we did not orient these relative to the origin of replication, and we were just sort of playing around with this, and we changed it to this by orienting them relative to the origin of replication. I don't know if you can see it on the side, but there's a clear X-like nature to this pattern. And what this means is basically that if you have a gene in Vibrio cholera, the distance that it is from the origin of replication is similar between Vibrio cholera and E. coli. Because these genes over this region over here, so here's a gene in Vibrio cholera, here's the origin of replication, it's 100,000 base pairs from the origin of replication, and it shows up on this axis. Or it shows up here. Up here is the other side of the circle. So if we're comparing E. coli to Vibrio, it may be hard to realize what this X means, but let's say we have a gene in Vibrio, gene A. The equivalent gene in E. coli may be here. If we have gene B, the equivalent gene in E. coli may be on the other side of the origin of replication, but it's the same distance from the origin of replication. Gene C may be here, gene D over here, and as we do this plot, on average, genes were the same distance from the origin of replication between E. coli and Vibrio, but the side that they were on was not conserved. Yeah, did you?
That's what I was gonna
Okay.
ask.
So we saw this, and I actually presented another version of this at a meeting. We hadn't oriented it to the origin and terminus of replication. And this, two people at the meeting got up after I said, I have no frigging idea what this means. And these two people, after I presented this, came up. This guy, Sean Eddy, who's a very famous bioinformatician now at the Howard Hughes Institute in Genelia Farm, and this E. coli guru, Monica Riley, both said, oh, maybe you have these symmetric inversions that pivot around the origin of replication, and that's why the distance that a gene is from the origin of replication is conserved. Actually, that's what Sean Eddy said. So he suggested this model where if you have an inversion that pivots around the origin of replication, the distance a gene is from the origin stays the same, but the side that it's on is not. Monica Riley, who was completely brilliant, dug out this old literature from E. coli that had proposed that E. coli had undergone this whole genome duplication and inversion of the duplication, where they suggested that sometime in the ancestry of E. coli, let's say you have, and so on, the whole genome got duplicated. And you ended up with A, B, C, D, E, and then another copy somewhere. And then there was some sort of inversion of this. And so, long story short, we don't need to go into all the detail of it. We tested these various models, and the only one that fit the data was the one that Sean Eddy had originally suggested, which was this pivoting around the origin of replication. And if you have, this is what this sort of dot plot shows. If you start with one genome, they diverge into two lineages, but they haven't changed any gene order. And you plot A1 versus B1, you see a perfect y equals x diagonal. If then between this point and that point, you get a little inversion that is symmetric around the origin, symmetric around the origin and symmetric around the terminus mean the same thing with a circle. And you do a dot plot of A2 versus A1, you would see one inversion here, right? Basically where the terminus is. The same thing happened here, B1 versus B2. You have an inversion here. If you now plot the descendants, A2 versus B2, you see this pattern. You add one more symmetric inversion in the A lineage and one more in the B lineage, and you plot them against each other, you're starting to see an X. And so, turns out that if you compare Mycobacterium tuberculosis versus leprae, you see a sort of X. Pyrococcus, different species, you see an X. Two different thermoplasma, which are two different archaea, you see an X. Bacillus stautilus versus Staphylococcus, you see an X. You can't quite see it, but in Streptococcus and Pseudomonas, there's an X. Every pair of closely related genomes that you look at sees something like this. It's completely, nobody expected this, basically. Although, nobody may have expected this to be a universal rule, but the reason Monica Riley had this model about ancient duplication in the E. coli genome was there were two groups in the, I think the 70s, may have been the 60s even, that started working on genetic maps of E. coli and Salmonella. And they noticed this weird thing about the position of the ribosomal RNA operons in these organisms, which appeared to be symmetric about the origin of replication. There were multiple copies of the ribosomal RNA, and they were spaced apparently symmetrically. And so these two groups both started working on this, and one of them said, maybe they're symmetric because there's selection against operons that are not symmetric around the origin of replication. And there's a reason for this, but we don't need to go into it. And the other one said maybe they're symmetric because inversions occur that are more frequently symmetric around the origin. One of these groups is actually John Roth, who's now a microbiology professor here. So they had been working on this for like 15 years in Salmonella and E. coli, developed the entire suite of ways to actually test for this. And nobody had realized that this occurs in every group of organisms, basically. And so, just to sort of finish this story, what they were looking at was basically this really interesting thing that happens in bacteria and archaea, and again is one of the fundamental differences that you might see with bacteria and archaea versus eukaryotes. You have a genome, and you have an origin of replication of this genome. Two really interesting things happen that are polar relative to the origin of replication. So one is that as this replication fork starts to grow, you're copying the DNA. The DNA polymerase is the DNA is getting pulled through this DNA polymerase. You can imagine the DNA polymerase is basically moving along the DNA. If you are trying to transcribe a gene while this is happening, and the gene is on the strand of DNA that the DNA polymerase is copying, if the RNA polymerase is facing the DNA polymerase, they're going to bump into each other. It's not good for the cell. If the RNA polymerase is making a transcript that's going away from the origin of replication, the cell will be happier. And it turns out that the orientation of genes relative to the origin of replication is highly biased in many bacterial genomes. And it's very clear that there is some type of selection against genes that are pointing towards where the DNA polymerase is coming.
And that's an asymmetric conversion.
Well, if you have, that's just in general, you don't want some genes, especially highly transcribed genes, you don't want to be opposite to the direction of the DNA polymerase. If you have a gene here, and you have a symmetric inversion, it actually gets flipped to the opposite side, and it will also end up pointing in the opposite direction of the DNA polymerase. If you have an asymmetric inversion, like here, it flips the orientation of that gene relative to the DNA polymerase.
Right now, that's what I was asking.
Yeah.
So those are that. So
This would be selection against other types of inversions because they change the orientation. And that's what some of these old Salmonella and E. coli experiments were working on. And there's another factor that's also important, which is that as this is happening, in fast-growing cells, you actually have many of these forks going around. And DNA that's near the origin of replication is present in more copies per cell than DNA that's near the terminus. It's called gene dosage. And as this is happening, you can, in E. coli, have 4 times as many copies of the DNA that's here as here. If you need to make lots of RNA off of a gene, or lots of protein, works better when you have 4 copies of that gene than if you have 1. So there's clear selection for proximity to the origin of replication because it determines your dosage. So again, symmetric inversions don't change the proximity relative to the origin. They don't change the gene dosage. Other inversions do. So there's this alternative model, though. This is the selection model. There's an alternative model which is also developed in these Salmonella experiments, which was you start replicating the genome. At any one point in time, the DNA polymerase is replicating DNA that is roughly equidistant from the origin of replication. If something happens, like DNA damage or the cell runs out of energy, the cell will stop. Maybe the DNA polymerase falls off or something. Maybe you get some strand breaks here. This could create inversions that now occur symmetrically around the origin because the DNA polymerase was equidistant from where it's replicating. Now, I mean, this is sort of a trivial, maybe relatively unimportant in the grand scheme of things, but this is an example of how the fact that bacteria and archaea in many cases have one origin of replication, or a small number of them, creates this polarity that leads to all sorts of other selective and comparative genomic sort of anomalies compared to eukaryotic genomes. This is just to show you sort of really quickly two last things about comparing genomes to each other. So if you line up one genome with another, this is one way to see examples of gene loss. If you to distinguish gene loss from genome insertion, if you have a tree, and you can infer that, say, the set of genes up here, which is what was done, these are basically E. coli genes, were present in all the ancestors because they're present in many different lineages, and then you have one group that's missing those genes. The simplest model is to infer loss of genes from that lineage. And that's what's occurred in many. So in bacteria, there's lots of cases of these gene losses as well as insertions, and you have to do some type of evolutionary analysis to distinguish which has occurred. Yeah.
So you just like kind of look for those chunks of DNA that have like exact matches in the same order. It's just
Yeah,
that they're
exactly.
a little bit spread out.
So you line up the protein coding genes in this case, and they're all in the same order between this. This is an endosymbiont of aphids. They're all in the same order as genes in E. coli. But just this organism is missing many of the genes present in E. coli. There are two possible explanations. They were added to E. coli or lost in this, and you distinguish them by then layering them onto a tree. And then we won't talk about it because we're out of time, but the last thing, big sort of genome change that occurs in many genomes is duplication of genes, and this is common, but much more common in eukaryotic genomes, at least sort of over long periods of time, probably because they can tolerate more extra DNA. But you still see it in bacterial and archaeal genomes. And we'll talk a little bit more about evolutionary analysis Thursday.